Good morning, all. Today we are here to discuss about the metabolism of carbohydrates. So, metabolism. What do you mean by metabolism? The entire spectrum of chemical reactions occurring in the living system are referred as metabolism. A metabolic pathway constitutes a series of enzymatic reactions to produce specific products. So, whenever a reaction happens, a chemical reaction, mainly it means a reactant which reacts to form a product or with the help of an enzyme. So here in biochemical reactions, all the biochemical reactions which involves an enzyme for its for the which catalyzes the biochemical reaction. So here the metabolism refers to the metabolic pathway, entire spectrum of chemical reaction occurring in the living system. Metabolism is divided into mainly two categories, that is catabolism and anabolism. Catabolism, the degradative process concerned with the breakdown of complex molecules to simpler ones with release of energy, that is known as catabolism. Catabolism means simply it is a breakdown of the molecules. Anabolism, the biosynthetic reactions involving the formation of complex molecules from simple precursors. From simple means inactive substances, the complex molecules are formed. That is known as anabolism. In simple words, we can discuss that metabolism is divided into two types, that is catabolism and anabolism. Catabolism is a breakdown of larger molecules, and anabolism is the simpler molecules which are fused together to form a new substances. So, carbohydrate metabolism. Here we are discussing about the metabolism of carbohydrates, how that carbohydrates gets degraded or how they are formed. So, carbohydrates are the major source of energy for the living cells. We all know that that our body needs a carbohydrate, which is a source of energy. So the monosaccharide glucose is a central molecule in carbohydrate metabolism, since all the major pathways of carbohydrate metabolism are connected with it. So here the center of attraction is the glucose, and from that all the molecules, all the carbohydrate metabolism are interconnected. So glucose is utilized as a source of energy. It is synthesized from non-carbohydrate precursors and stored as glycogen to release as glucose when needed. So here glucose is a source of energy and it is synthesized from the non-carbohydrate precursors and it is stored as glycogen, which in future, if we are in fasting, this glycogen is used as a glucose, or it serves as a an energy source for the body. So the other monosaccharides important in carbohydrate metabolism are fructose, galactose and mannose. So these are the major pathways of carbohydrate metabolism. First comes the glycolysis. It is the oxidation of glucose to pyruvate and lactate. That is, it is a catabolism. That means the glucose is split into pyruvate and lactate. Then second one is citric acid cycle. It is also known as TCA cycle or Krebs cycle. So that it is a oxidation of acetyl CoA to carbon dioxide. The next one, gluconeogenesis, that where the name states that it is a formation of new molecules of glucose, the synthesis of glucose from non-carbohydrate precursors. Then glycogenesis, here the name suggests what genesis of glycogen, that is the formation or synthesis of glycogen from glucose. The next one, glycogenolysis, where the breakdown of glycogen to glucose. Then sixth pathway is hexose monophosphate shunt or HMP shunt. It is an alternative pathway to glycolysis and citric acid cycle for the oxidation of glucose. Uronic acid pathway. Glucose is converted to glucuronic acid, pentoses and ascorbic acid. An alternative oxidative pathway for glucose. Then comes galactose metabolism, conversion of galactose to glucose and the synthesis of lactose. Then the ninth one is fructose metabolism, oxidation of fructose to pyruvate. Then the 10th one is amino sugar and mucopolysaccharide metabolism, the synthesis of amino sugars and other sugars for the formation of mucopolysaccharides and glycoproteins. Of these carbohydrate metabolism, the important metabolisms are first we are going to discuss mainly about these six metabolism, that is glycolysis, citric acid cycle, gluconeogenesis, glycogenolysis, hexose monophosphate shunt. These are of more important. So, coming to the point, pathways involved in the intermediary metabolism of glucose. So, here we have already discussed that means metabolism is of two types, anabolic and catabolic. So, here anabolic pathways are glycogenesis, glucogenesis, lipogenesis. Then catabolic pathways means glycolysis, glycogenolysis, oxidation of pyruvate to acetyl-CoA, HMB-shunt and uronic acid pathway. These all are catabolic because these are the breakdown of larger molecules into simpler molecules. And in anabolic pathways, there is a formation of new molecules. That's why these are the examples for anabolic pathways. And TCA cycle is the only cycle which is an example of both amphibolic pathway, which means this is a reaction. This is a cycle where both anabolism and catabolism takes place. So next one, compartments of different metabolic pathway. Mainly all the biochemical reactions, all these cycles takes place either in the cytosol or in the mitochondria. So here we are going to discuss what are the reactions takes place in the cytosol, mitochondria and in endoplasmic reticula. So in cytosol, glycolysis takes place, HMB shunt takes place, glycogenesis, glycogenolysis, lipogenesis, transamination takes place. And in mitochondria, mainly TCA cycle, RC oxidative phosphorylation, beta oxidation, ketogenesis, deamination takes place. In both the cytosol and mitochondria, some of the reactions took place. They are gluconeogenesis, urea cycle, and heme synthesis. Endoplasmic reticulum, there happens the protein synthesis and the cholesterol synthesis. So this is the metabolic pathways. So first we are going to discuss about the glycolysis. In this whole slide we are going to discuss about what is the glycolysis and what are the process involved in, how much energetics, that how much energy is produced, how much ATPs are produced, that is known as energetics, and what are the significance of this cycle, that we are going to discuss about in this whole slide. So first slide, glycolysis. The other name for glycolysis is known as Embden-Meyerhof pathway, EMP pathway. That means this cycle, this metabolism is put forward by these two scientists, Embden and Meyerhof. That's why the name is given as Embden-Meyerhof pathway. Glycolysis is defined as a sequence of reaction converting glucose or glycogen to pyruvate or lactate with the production of ATP. Glycolysis takes place in all cells of the body. The enzymes of these pathways are present in the cytosol fraction of the cell. Glycolysis can be either anaerobic or aerobic. Lactate is the end product under aerobic and anaerobic condition, and in aerobic condition the pyruvate is formed, which is then oxidized to carbon dioxide and water. This is the overview for glycolysis or an introduction for glycolysis process. So glycolysis, oxidation of glucose to pyruvate or lactate with the production of ATP by Embden-Meyerhof pathway is called glycolysis. There are two types of glycolysis, aerobic glycolysis and anaerobic glycolysis. In aerobic glycolysis, there is a presence of oxygen and it is converted into pyruvate. In anaerobic glycolysis, it is converted to lactate. That is the only difference. The end product will be differed in aerobic and anaerobic glycolysis. So, coming to the point, aerobic glycolysis, breakdown of glucose in the cell with mitochondria. Inadequate supply of oxygen is called aerobic glycolysis. The end product of aerobic glycolysis is pyruvate, which is then converted to acetyl-CoA and enters into the TCA cycle, to which it is converted to the carbon dioxide as an end product. So, this is the end product of the glycolysis, which will be the starting material for the next cycle, which is continued by the TCA cycle. So anaerobic glycolysis, breakdown of glucose in the absence of oxygen, is called anaerobic glycolysis. Occurs in cells without mitochondria. Pathway is same as aerobic except end product. So just now I have told that the end product will be differed in this aerobic and anaerobic. Rest all the reactions are same. So here it occurs in RBC, brain, gastrointestinal tract, testis, renal medulla, skin, skeletal muscles and retina. So the pathway of glycolysis, what are the salient features? Substrate, the main product which starts the chemical reaction, which starts the biochemical pathway, which includes glucose or glycogen. And the product is pyruvate in aerobic, that is the presence of oxygen, and lactate in the absence of oxygen, that is anaerobic. Site almost all tissues and cells. Compartment, cytoplasm. The enzymes which are involved in these reactions are mainly present in the cytoplasm. The nature, what is the nature or what is the type of metabolism here take place means it is a catabolism, that is a breakdown. So steps here, we have seen the flow chart for the reaction. Here it is given that glucose is converted to glucose 6 phosphate in the presence of hexose kinase, where it utilizes ATP, which is converted to ADP. And again glucose 6 phosphate is converted to fructose 6 phosphate with the help of an enzyme isomerase, phosphoglucose isomerase. And again it is converted to fructose 1,6 bisphosphate with the help of a enzyme phosphofructokinase, and it utilizes one more ATP molecule. Again it is converted to glyceraldehyde phosphate and dihydroxyacetone phosphate. Here dihydroxyacetone phosphate again, which is rejoined to form 1,3 bisphosphoglycerate, 3 bisphosphoglycerate, 2 phosphoglycerate, phosphoenol pyruvate, and then pyruvate with the help of following enzymes that we can discuss in the upcoming steps. Reaction of glycolysis. Reactions of glycolysis. The pathway is divided into 3 distinct phases. First one, energy investment phase or priming stage. Second one, splitting phase. Third one, energy generation phase. In these 3 phases, first energy investment phase, where the energy is utilized, the energy is used or consumed. And the third step, energy generation phase, where the energy is produced. So here we have already seen that in the first 2 steps, first 3 steps, first and third step, there will be a usage of ATP which is converted to ADP. So here the energy currency, that is ATP, is utilized. That's why it is known as the energy consumption or energy investment phase. So first one, we are discussing about the first phase. Glucose is phosphorylated to glucose 6 phosphate by hexokinase or glucokinase. This is an irreversible reaction dependent on ATP and magnesium 2+. Here the magnesium ion and ATP is dependent, and it is an first step is an irreversible reaction where the glucose is converted to glucose 6 phosphate. And the second step, glucose 6 phosphate undergoes isomerization to give fructose 6 phosphate in the presence of enzyme phosphohexokinase, hexose isomerase, and magnesium ion. Third step, fructose 6 phosphate is phosphorylated to fructose 1,6 bisphosphate by phosphofructokinase, irreversible and a regulatory step in the glycolysis. This third step is a regulatory step in this whole glycolysis process. Then comes the splitting phase. So here the first 3 steps which are involved in the energy investment phase or priming stage, and the second comes the splitting phase. Here the 6 carbon fructose 1,6 bisphosphate is split to 3 carbon compounds, glyceraldehyde 3 phosphate and dihydroxyacetone phosphate by the enzyme aldolase. By the enzyme aldolase, this fructose 1,6 bisphosphate is converted to dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. So the enzyme phosphotriose isomerase catalyzes the reversible interconversion of glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Thus, 2 molecules of glyceraldehyde 3 phosphate are obtained from 1 molecule of glucose. So here from 1 molecule of glucose we obtain 2 molecules of glyceraldehyde 3 phosphate. Next comes the energy generation phase. Here glyceraldehyde 3 phosphate dehydrogenase converts glyceraldehyde 3 phosphate to 1,3 bisphosphoglycerate. NADH and H and a high energy compound 1,3 bisphosphoglycerate is formed. Iodoacetate and arsenate inhibit the enzyme glyceraldehyde 3 phosphate dehydrogenase. In aerobic reaction, the NADH passes through the electron transport chain and 6 ATP are synthesized. 23. ATPs are synthesized. The enzyme phosphoglycerate kinase acts on 1,3 bisphosphoglycerate, resulting in the synthesis of ATP and formation of 3-phosphoglycerate. Again, the 3-phosphoglycerate is converted to 2-phosphoglycerate by the enzyme mutase. This is an isomerization reaction, and the high energy compound phosphoenol pyruvate is generated from 2-phosphoglycerate by enolase. Enolase, with the help of an enolase enzyme, the pyruvate is formed. Enolase requires magnesium and manganese and is inhibited by the fluorides. The enzyme pyruvate kinase catalyzes the transfer of high energy phosphate from phosphoenol pyruvate to ADP, leading to the formation of ATP. Pyruvate kinase requires potassium and either magnesium or manganese to form the ATP. So that's why in the last reaction, last phase, it is known as energy generation phase, where the ATPs are formed. Then the next slide explains about what the triads glucokinase and hexokinase. That means the difference between the enzymes glucokinase or hexokinase. Affinity for glucose, glucokinase have low affinity, hexokinase have high affinity. Therefore, in low affinity, the Km value, that is Michaelis-Menten constant value, is higher, and in hexokinase, Michaelis-Menten value is lower. Specificity, phosphorylated only glucose. That hexokinase is non-specific, can be phosphorylated any hexose. Location, it is mainly present, glucokinase is mainly present in liver cells and pancreatic islet cells. Hexokinase is present in extrahepatic tissues. Action of insulin, glucose it can be stimulated. Action of insulin can stimulate glucokinase, not affected. Action of insulin will not affect the hexokinase. Action of glucose, not affected, allosterically inhibited in hexokinase. Then the net effect, remove glucose from blood following meal. In glucokinase, hexokinase maintain blood glucose concentration. So then the regulation of glycolysis, hormonal regulation, insulin, glucagon. These two are the opposite hormones, and the insulin increases the activity of enzymes in the liver, that is glucokinase, phosphofructokinase and pyruvate kinase, increases the conversion of glucose to pyruvate. That is a function of insulin. That's how the glucose levels in the blood are decreased by the insulin. The insulin will increases the conversion of glucose to pyruvate. Thus, the levels of glucose is decreased. So, in glucagon, opposite effects in fasting and in diabetes mellitus. Inhibit the activity of glucokinase, phosphofructokinase and pyruvate kinase. This have the exact opposite action. Then regulation of glucose. In well-fed state, increased ingestion of glucose, increased blood glucose, increased release of insulin, increased protein phosphatase activity. In fasting stage, increased level of glucose, increased glucagon, increased CMP, increased protein kinase activity of enzyme. This is the regulation of glycolysis. Then energy production. Here we have already said this is the energetics, how much ATPs are formed, how much energy is generated. That's what here we are discussing. In aerobic, in respiratory chain, 5 ATPs, at substrate level 4 ATPs. Net gain 9 minus 2 is equal to 7 or 8 ATPs. That is, in aerobic condition, the ATPs formed are up to 6 to 8 molecules of ATPs are formed. In anaerobic condition, only 2 ATPs are formed. Rest are utilized in the utilized by the further steps in the anaerobic reactions. Then importance of glycolysis. Only pathway in all cells of the body. Only source of energy in RBC. Then in strenuous exercise, major source of energy. Allow tissue to survive anoxic episodes. Then deficiency of enzymes of glycolysis, example pyruvate kinase, leads to diseases hemolytic anemia. So importance of glycolysis means it is the only pathway in all cells of the body. So and also it is a source of energy which is helpful for means almost all the biochemical reactions. Then preliminary steps for the complete glucose oxidation. The second one is reversible steps are used for gluconeogenesis. in glycolysis, we have steps which are the which is splitting up of glucose to pyruvate or lactate. But in gluconeogenesis, we have the reverse mechanism. So that is, in the name itself, it states that it is a formation of new molecules of glucose. So from reverse, it will be formed. Provides carbon skeleton for synthesis of non-essential amino acids and glycerol, part of fat. Then fate of pyruvate. In cytoplasm, lactate, which helps in reduction, alanine transamination, glucose gluconeogenesis. In mitochondria, acetyl-CoA, oxidation and decarboxylation. Oxaloacetate carboxylation. So in here, the acetyl-CoA, which is the starting molecule for the citric acid cycle, and the oxaloacetate is regenerated. Then source of pyruvate. The sources of pyruvate are from glucose, which is glycolytic pathway, amino acid from transamination, glycerol part of fat, etc. Here, this diagram shows what means how this pyruvate is produced from malic enzyme, which releases pyruvate, from glucose glycolysis, which the end product is pyruvate, from lactic acid lactate dehydrogenase, which forms pyruvate, and from alanine ALT or SGPT, alanine transaminase, which forms pyruvate. This pyruvate is later converted to oxaloacetate or acetyl-CoA. Oxaloacetate by pyruvate carboxylase and acetyl-CoA by pyruvate dehydrogenase complex. Thus, this pyruvate is synthesized and it is converted. Oxidation of pyruvate to acetyl-CoA. The oxidation to pyruvate to acetyl-CoA is the irreversible route from glycolysis to the citric acid cycle that I have discussed earlier. So, here the pyruvate is formed, which is the end product of aerobic glycolysis, and it is the starting product for the citric acid cycle, which enters into the citric acid cycle. Pyruvate is transported into the mitochondria by a proton importer. If it then undergoes oxidative decarboxylation to acetyl-CoA, catalyzed by multi-enzyme complex, pyruvate dehydrogenase complex present in the inner mitochondrial membrane. Inhibition of pyruvate metabolism leads to lactic acidosis. So, oxidation of pyruvate to acetyl-CoA, salient feature, substrate here it is pyruvate, product is acetyl-CoA, site all tissues, compartment mitochondria, nature catabolic. Here the oxidation of pyruvate to acetyl-CoA is given. Here the pyruvate is transported to the mitochondria with a symport, proton importer. So that's how this pyruvate is entered into the mitochondria from the cytosol, and it is converted to the acetyl-CoA with the help of an enzyme. Pyruvate dehydrogenase complex. Here the multi-enzyme, which is a multi-enzyme complex associated with inner mitochondrial membrane, analogous to alpha ketoglutarate dehydrogenase complex of citric acid cycle. It requires 5 enzymes: TPP, vitamin B1, coenzyme A, NAD, FAD, and lipoic acid. So that's why the pyruvate dehydrogenase complex is a complex, multi-enzyme complex. So that's all for today. The carbohydrate metabolism, the first cycle, that is glycolysis, we have elaborately discussed about that. First, in this shortly we can have a feedback, means review. First, we have discussed about the what is metabolism and what are the types of metabolism, and coming to the next part, we have discussed what are the reactions, what are the metabolic pathways which is involved in the carbohydrate metabolism. And of that, first one we have discussed in this whole slide, that is glycolysis. It has another name of Embden-Meyerhof pathway, EMP pathway. and it is a main pathway for all other um, metabolism. It is a main pathway for all which takes place in all the cells. And we have discussed what are the location, what are the enzymes present in that, and what are the steps in this glycolysis, and what are the end products. So here in anaerobic reaction, two means criteria occurs, and in aerobic reaction, the end product formed from the glucose is pyruvate, and in anaerobic reaction, that is in the absence of oxygen, the end product formed is lactate. So we have discussed the steps. What are the steps involved in that? What are the uh, what are the enzymes involved in that? So the whole step in the glycolysis process is categorized into three steps. That is, first one is the energy investment phase, second one is splitting phase, third one is energy producing phase. So that's all from that. And then we have discussed about what are the salient features, what are the importance of significance of this glycolysis, and what are the energetics. So that's all for today.